R. W. Holley is known for his research in RNA and protein synthesis and is accredited with sequencing the tRNA molecule. Born on January 28, 1922, in Urbana, Illinois, Holley was one of the four sons of Charles and Viola Holley, both of whom were educators. In 1945, he married Anne Dworkin and later had a son named Frederick. Holley died on February 11, 1993, at the age of 71. Holley attended Urbana High School, received a Bachelor of Arts degree from the University of Illinois in 1942, and received his Ph.D. in organic chemistry in 1947 under Professor Alfred T. Bloomquist. Because of World War II, Holley spent 2 years researching under Professor Vincent Duvignean, shown here, at the Cornell University Medical College. There, he participated in the first chemical synthesis of penicillin. In 1948, Holley became an assistant professor of organic chemistry at Cornell and was later appointed a professor of biochemistry. From 1955 to 1956, Holley was on sabbatical, studying with James F. Bonner at the California Institute of Technology. It was here that Holley first began researching protein synthesis. In 1968, Holley was awarded the Nobel Prize in Physiology and Medicine, along with Har Gobind Khurana and Marshall Warren Nierenberg. They were selected for describing the structure of alanine transfer RNA, thereby connecting DNA and protein synthesis. Holley's research consisted of two main goals: isolating transfer or tRNA and determining the sequence and structure of alanine tRNA. After isolating tRNA, Holley used two ribonucleases to break the nucleic acid tRNA into small pieces. The ribonucleases in question were pancreatic ribonuclease and tachydiastase ribonuclease Ti. Ion exchange chromatography and paper electrophoresis, or rechromatography, were used to isolate the fragments of RNA. In this diagram from Holley's Nobel lecture, we see how these methods can be used to determine the sequence of the fragments. By hydrolyzing the fragments with alkali, the mononucleotides could be identified using chromatographic and electrophoretic properties, as well as spectra. The cleavage site of the ribonucleases was known, so the dinucleotide sequence of the tRNA could be determined. By 1964, the alanine tRNA molecule was completely sequenced. Holley and the other scientists continued to research the structure of tRNA and its use in translation. They observed the cloverleaf structure and the anticodon sequence, which binds to the complementary codon on the mRNA strand. Here we see Holley attempting to determine the structure as well as the sequence of the alanine tRNA nucleic acid. In later years, Holley's method was used by other scientists to sequence other tRNA molecules. The same method was then used to sequence nucleotides in bacterial, plant, and human viruses. Holley, meanwhile, became the resident fellow at the Salk Institute for Biological Studies in La Jolla, California. Holley's success was very much driven by his personality. Holley's main interests included mountains and oceans. His interest in natural areas could indicate that he wanted to see new areas. He would thus persevere to see new natural areas, like he would persevere to fully sequence the tRNA molecule. Since both of his parents were teachers, he most likely grew up in a household that fostered an interest in learning, compelling Holley to become intelligent and have a natural curiosity. By continually researching protein synthesis for almost 10 years, Holley shows a high level of diligence. Earning a Ph.D. in biochemistry indicates that Holley was dedicated to the field, as well as to research and discovery. This shows that he was willing to put in the time and effort to sequence tRNA and determine its structure. Holley was awarded the Nobel Prize in Physiology and Medicine in 1968 for his interpretation of the genetic code and its function in protein synthesis. His life experiences led him to be successful in his research and future endeavors. I think that Holley's discovery was incredible, as it allowed us to learn about how DNA was translated into an amino acid sequence, as shown here. His personality traits portray him as a dedicated, passionate man who was interested in learning and discovery, and I believe that his research further opened the door to genetic engineering. The significance of his research is best expressed by this quote from the speech at the presentation of the 1968 Nobel Prize. "We can now begin to understand the causes of many diseases in which heredity plays an important role."